Hey everybody, what's going on, right? I somehow just scared my dog, and I don't know if you can hear her barking. I just got so into that intro, right? Take a minute and chew on that. Ha! I hate myself. All right, so digestion and absorption. The past couple of weeks, what we've been focused on is macronutrients and micronutrients. Now we're kind of putting it all together in some way, shape, or form. I'm trying not to come up with a really bad pun right now about how this all breaks down, right? But this is kind of where it all starts going down. So up until this point, we've talked about what the macronutrients do and what the micronutrients do. This lecture, we're really going to be focused on how do we get the nutrition and the sustenance out of it. So with that being said, what is digestion, right? So digestion is going to be a two-part process. It's going to be both mechanical and chemical breakdown. So the mechanical is going to be the mastication. It's going to be the actual chewing breakdown. We're also going to have some jostling around that's going to occur within the stomach, kind of help mix some stuff together, and we're going to have some mixing that kind of continues to occur as we go through. We're also going to have that peristalsis action, this rhythmic smooth muscle contraction that's going to help kind of flow stuff through our system. We're going to be able to then absorb, as we're kind of pushing stuff through our system, moving stuff through our system, we're going to be able to absorb those nutrients across the intestines and put them into our body where we need them. Something that we kind of just in passing in the last literally 30 seconds of class on Monday talked about is that the insides of you are technically on the outside of you. Something to kind of think about is it's one continuous path from your mouth all the way down to the anus. So there is literally, it's like it's on the outside of you, even though it's on the inside of you. Kind of a little bit goofy. So we're constantly trying to get nutrients into our body, even though they're already in our body. Right? So what is it that's going to really kick off digestion? Well, think about what it is and what happens when you smell something like pizza or you smell fresh baking cookies. You kind of go ahead, you start to salivate, you start thinking about that food, your stomach, you're like, oh man, maybe I am kind of hungry. And that's really going to be what's kicking off the digestive tract. That first thing is going to be that sense of smell. And then once we kind of put that food into our mouth and we start to get that chewing and we get that smashing, that's going to also trigger some of those digestive responses to happen. Now, inside of the mouth, we're also going to have things like salivary amylase and salivary lipase that are going to start breaking down carbohydrates and start breaking down some of these fats so that we can actually get access to them as we move through. So one of the first things that we're going to have to do if we're trying to take a cheeseburger from the environment and put it into our body and break it down into carbs and fats and protein, we're going to have to take and break down that cheeseburger. We can't just absorb a whole cheeseburger into us. Same thing, regardless of what we're eating, right? We're going to have to mechanically break it down for some way, shape, or form. So that's going to be using our tongue to move it around inside of our mouth. It's going to be chewing on it to break it into progressively smaller pieces so that we're trying to actually increase the surface area of that product. The reason we want to increase that surface area is because we need enzymatic breakdown to occur. Enzymes can only work on the surface level, so they can't get into the middle. The best way to increase surface area of something is to break it down into a million different pieces, right? Break it down into small little pieces. We're then going to kind of take and push all this together and throw it down the back of our throat so that it can hit our stomach. Realistically, we're going to have 4 primary hormones that we're going to be talking about throughout the rest of this lecture and kind of throughout the rest of the week, right? We're going to be talking about gastrin, gastric inhibitory peptide, secretin, as well as cholecystokinin. I'm not going to sit here and read to you guys everything that's on this slide, but suffice it to say that each one is going to do something different, right? Each one is going to have its own independent role. Cool enough, CCK actually we'll talk about a little bit more later, but it has direct coordination with the brain. So this is kind of where we start getting some gut-brain axis and some gut-brain coordination to start talking about how full we are. So it might actually stimulate some of that fullness. Where are some of these hormones going to be coming from? Where are these digestive secretions going to be coming from? Well, we're going to have them starting in the mouth, right? We're going to have salivary amylase and we're going to have salivary lipase. So it's going to be produced in the salivary glands. We're going to have gastric glands. Our gastric glands tend to be in our stomach, hence gastric bypass. Anytime we're really using the word gastric, we're talking about the stomach itself. We're going to have the pancreas is going to be secreting stuff. The liver, the gallbladder, the intestinal glands are going to be secreting stuff. Each one of these secretions and each one of these organs and target organs is going to have something specific about it, right? So the salivary glands are going to have a target of the mouth, right? Salivary, salivate. So we're going to try to start breaking down foodstuffs inside of our mouth. Take, for example, another one would be the liver, right? An organ that is the liver is going to actually be the thing that produces bile. We're going to store that bile within the gallbladder, and whenever we consume fat, we will secrete that bile in order to try to emulsify that fat. So each one of these things is going to do something different, right? Up to this point, we've gotten the nutrients into the system. We're through the stomach, we start having some secretion, whatever, right? How do we actually get things across the cell? Well, just like everything else inside the body, it's all going to be trapped within cells. So everything is going to be held within cells. How do we take something from outside of the body, even though it's on the inside, but it's on the outside, but it's on the inside, but it's on the outside? How are we going to get these things from the intestinal tract and get them into our body? Well, we're going to have two primary transport mechanisms. We're going to have passive transport and we're going to have active transport. Now, passive transport means that there's going to be no external energy being required. Active transport means that we're going to actually have to use energy in order to power the transport. In order to actually get the absorption to occur, we have to use ATP. We have to use energy in order to do it. So, two primary types of transportation, passive and active. When it comes to passive transport, we're going to have a couple different types of passive transport. One example is going to be simple diffusion. It's how water is going to actually spread across membranes. Another one is going to be facilitated diffusion. We're going to actually use like specific transporters and use specific channel proteins in order to move through the cell membrane. Another good example is going to be osmosis. And osmosis is simply the travel of water across these semi-permeable membranes. Active transport gets a little bit different. We're going to have a couple of different types of active transport. One primary one is going to be sodium potassium pump. For those of you guys who have already passed anatomy, which should be all of you, sodium potassium pump is going to be used for maintaining ion gradients and maintaining molecule gradients across the nervous system as well as within the muscle. And realistically, what we're doing here is we're treating this like an exchanger. It's actually an exchange mechanism. So we're going to be exchanging sodium for potassium. We're going to be exchanging 3 sodium for 2 potassium, trying to basically maintain a specific gradient of positive ions. Another example of active transport is going to be coupled transport, which means that we're going to symport things. We're going to actually move them together. We're going to use some sort of carrier protein to move them together through the membrane. And then we're going to have bulk transport. Bulk transport, think about it like we're grabbing something in bulk, right? Like we're pulling something big in or we're blowing something big out. So let's talk about each one of these a little bit more. Sodium potassium pump is all about ion regulation. Here what we're trying to do is we're trying to maintain positive chain, like we're trying to maintain the positive ions. When we are in a resting state, like when we are in a resting membrane potential state, when we are just hanging out, we need to be maintaining about negative 70 millivolts. So that means that the outside of the cell is going to be higher positivity than inside the cell. When we depolarize, we actually have an increase in positivity until we hit negative 55 millivolt. Once we hit negative 55 millivolt, we're going to amplify that signal and it is going to go extremely positive. Once it hits extremely positive, it's going to actually flip around and repolarize all the way back down to super negative. The way that we repolarize is just by changing out the positive ions. So we allow for more positive ions to leave the cell than we bring back into the cell. Overly complicated. If you haven't had phys yet, don't stress about it. There's not going to be huge amounts of questions about this. Just know that it is an active transport mechanism. Now, coupled transport is going to be where we're actually using some sort of a sym-porter protein. And now sym-port means that it's both going in the same direction. There is something called an antiporter, which means that they go opposite directions. Sym-porter, they both go the same direction. So here, this one is going to be a glucose and sodium sym-porter. So it's actually going to grab and transport that sodium and that glucose through the cell and into the target destination. And then that bulk transport is going to be responsible for exocytosis and endocytosis. Exocytosis, think about this is how we actually release our neurotransmitters when we're at the axon terminals, right? So we're actually going to have this transport vesicle that is just plasma membrane. All that it is an acting like plasma membrane because it is plasma membrane. It is going to be full of whatever it is we're trying to release. That plasma membrane is going to actually go up and bond and become one with the regular plasma membrane, the outer cell membrane. When it does that, it opens up a channel so that we can release all of the things that are held within that transport vesicle, right? Kind of efficient, kind of cool to think about how this is working. Think of it like you're taking a trash bag and then dumping the trash bag, or you're taking a recycling bin out to the street and then dumping it in a big recycling bin. My father is the worst about this. He will take and dump the recycling straight into the trash. I don't know. He doesn't believe in recycling. Endocytosis. This is going to be the complete opposite. Rather than having the transport vesicle fuse with the membrane and release things to the external environment, we're actually going to be engulfing things in the outside environment by pinching in the cell membrane. So this will actually pinch in right here and end up connecting and forming a new baby cell, essentially, within the other cell. Kind of a very efficient way to transport large things. Technically, we have two different types of endocytosis. We have phagocytosis, which means that we're consuming something. Our cell is consuming something solid, or we have penocytosis, which means that it's drinking. As the great Mahatma Gandhi would say, eat it or yeet it. So moving on along with GI tract anatomy, where this is going to start with the mouth and the esophagus. This is where that initial side of mechanical digestion is really going to occur with the goal so that we can actually fit stuff down our throat and so that we can increase the surface area of the product. Once we move that past the pharynx, once we move that kind of past the mouth, it's going to hit the esophagus. It's where we're going to actually have this powerful rhythmic smooth muscle contraction to actually squeeze that food all the way down and hit the stomach. So here's a fantastic overview of what the digestive anatomy looks like. We're going to start with the mouth. Inside of the mouth, we're going to have salivary glands. We're going to move food down through the esophagus. We're going to move that food from the esophagus through the cardiac sphincter and into the stomach, right? Once we get into the stomach, we're going to move things past the pyloric sphincter and into the small intestine. Once we're in the small intestine, we're going to move through the small intestine until we eventually get all the way into the ileocecal valve. When we get through that ileocecal valve, we're going to be in the large intestine. And then that large intestine is going to be the final kind of stopping point and final site of absorption. This is where we're going to get a lot of our minerals and water absorption coming from, right? So one more time, we're going to start in the mouth. We're going to move past the pharynx, down the esophagus, through the cardiac sphincter, into the stomach. From the stomach, we're going to actually move through the pyloric sphincter and into the small intestine. We're going to move through the small intestine, and we're going to go through the ileocecal valve into the large intestine. We'll then work through the large intestine and eventually exit the body. So it seems like a lot, but realistically, what are our sites of digestion? We've got the mouth. We have some stomach, not a whole bunch, small intestine, large intestine. So it really pares down, when you're looking at the big picture, really pares down pretty good. So let's talk about the stomach, right? Yes, the mouth and the esophagus is technically a part of this, but the stomach is really the first thing that we think of when we really start thinking about digestion. Outside of the really neat fact that it can expand like 6 or 7 times its normal resting size, it does a lot of really good things, right? Inside of the stomach, we're going to produce and secrete hydrochloric acid. This hydrochloric acid is crazy effective. It is super, super, super acidic. And it's going to help kind of stimulate some of the breakdown of the foods that we just kind of ate, right? We're going to also produce pepsinogen, which is going to be an inactive form of protein digesting enzyme pepsin. And we're going to just kind of straight move carbohydrates through here. Realistically, there's not a whole bunch of carbohydrate digestion or absorption that's going to be occurring inside the stomach. Most of that's going to be in the small intestine. So what is the stomach really good for? Well, it's really good for mixing everything together and storing everything as we start to move it through our small intestine. It will kind of get the party kicked off, but a lot of this is going to occur as we move into the small intestine, right? So 90% of lipid digestion is going to be occurring, essentially 90% of all digestion and almost all of lipid digestion is going to be occurring here in the small intestine. And that's only in the first 2 sections of the small intestine. That is wild, right? Almost all of our digestion is coming from a small intestine. And the way that it's going to do this is it's actually going to use villi and microvilli. So we're actually going to use this like brush border, this kind of like sweeping action in grabbing those nutrients and absorbing them through those microvilli and villi to kind of get the good stuff out of what we just ate. So let's kind of look at what that looks like. So we'll get past the esophagus, past the stomach, and we'll move into that small intestine, the first 2/3 of that small intestine. And we'll see that we have these intestinal mucosa. So we kind of have these brush finger-like extensions coming off of the small intestine wall here. If you look, basically what's going on here is we've got a series of zoom ins, right? Like zoom, 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 right? Zoom, zoom, wait, zoom, 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 zoom. Yeah, zoom, 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 zoom. I don't know. Once we get in here, we're going to take and zoom one more time in. And what I want you paying attention to is that we actually have these really small microscopic microvilli that are going to be acting as projections off of the regular villi. Now, the reason that we're going to do this is because it's dramatically increasing the surface area and dramatically decreasing the diffusion distance. So once we get past the small intestine, we're going to move into the large intestine. That large intestine is the final digestive structure. And realistically, there's no villi present. So we're not going to have that really cool brush border. We're not going to have this really small absorption distance to cover. What we're going to have is bacteria and yeast. So we're going to actually be fermenting things in here. This is going to be our method for absorbing water, absorbing electrolytes, minerals, things like that. But it's also going to be where we're fermenting and actually providing some of the butyrate that our colonocytes need in order to survive. Now, this is a really cool figure, I think, because it does a really good job of breaking down all of this into one kind of digestible. I'm here all week. Kind of one really good bite size. All right, I'm done. Into one really good figure where you can kind of see what's going on with carbohydrate, protein, and lipid. You can see the various places that it's going to be digested and how it's going to be absorbed, and then what vessel it's actually entering into. Something that's really kind of neat to think about is that lipids, for the most part, are going to be actually transported through the lymph into the blood first. There's a caveat here. Our medium chain triglycerides and our short chain fatty acids are actually going to go straight to the liver, where they're going to be converted into ketones and then released into the blood. But for the most part, lipid kind of gets absorbed straight into the lymph and then into the blood. So it's in the lymph that we're actually going to be producing and kind of generating some of those lipoproteins that we talked about last week. And if you look, you can see that the protein gets hit with some of the acid and the pepsin from the stomach. And then we're also going to have trypsin and other enzymes coming in to break everything down into single amino acids, where we're going to absorb it into the blood and send it straight through the liver, eventually back to the heart and into the systemic circulation. Carbohydrate is going to pretty much bypass the stomach, right? Like, you're not seeing a whole bunch of stuff going on inside the stomach, But we have salivary amylase occurring in the mouth. We have pancreatic amylase, and we've got all those brush border enzymes that are going to be breaking things down so that we can eventually have that get broken down into blood sugar or into glucose to be put into the blood and then either be stored as glycogen in the liver or in the muscle, or stored as blood sugar or put into the cells. And then you can see fiber down there in the large intestine, right? All the way down here in this bottom corner. It's going to be fermented to create it's going to be fermented with bacterial enzymes, and it's going to produce butyrate, which is a form of gas. It's also going to refer like flatulence, right? Ha 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 ha, gas. I don't know. I'm in a weird mood, guys and girls. But let's talk a little bit more about carbohydrate digestion. So the previous slide was generally what you need to know. Let's talk about it a little bit more in depth. So that carbohydrate digestion is going to start in the mouth. We're going to release salivary amylase. So if you put sugar in your mouth, the first thing that's going to happen is it's going to get hit with that salivary amylase. That food mixture is going to move through to the stomach. When it hits the stomach, there's going to be some breakdown. It's not completely without a job, but there's not going to be a ton going on. It's going to move into the small intestine. In that small intestine, we're going to use pancreatic amylase to basically break the starch into glucose and other monosaccharides. And then at the microvilli border within the small intestine, we're going to have a breakdown of maltose with maltase, sucrose with sucrase, and we're going to have a breakdown of lactose with the enzyme lactase so that we can finalize the breakdown into these monosaccharides. There, we're going to actually pull everything out of the system, put it into circulation so that it can end up where it's going. Lipids are not completely dissimilar from carbs, relatively similar. If you think about the structure, it kind of makes sense, right? The major difference here is the oxygen and the hydrogen to carbon ratio. So there's going to be quite a bit of similarities. The big difference here is that sugar and carbohydrates are dissolvable in water, whereas fat is not dissolvable in water. So we have to separate it out somehow. We have to emulsify it somehow. That's the major difference, right? So when we consume lipid, it's going to start with lingual lipase. We're going to, the salivary lipase, lingual lipase, whatever. We're going to start the digestion here. We're going to start the breakdown of lipids actually inside of the mouth. As we move into the stomach, we're going to have continued breakdown of triglycerides. And then we're going to get into the small intestine where it's like virtually all of lipid digestion is going to be occurring, right? Large majority of it is going to be occurring. So how is that going to be happening? Well, one of the major things that's going to happen is cholecystokinin, CCK, is actually going to be released. When this CCK gets released, it's also going to stimulate for gallbladder contraction and bile flow. So we're actually going to release bile. This is so important. This is so big because we have to be able to emulsify that fat. If we just pour oil into water, if we just pour oil into these liquid substances, what's going to happen is it's going to stay together and it's not going to be able to get broken up as easy. What bile salts do, what bile does, is it gets in there and emulsifies it. So it turns it into hundreds of little micelles, all these little bubbles, so that we actually have an increased surface area so that the enzymes can hit it to start breaking it down and so that we can ease the absorption somewhat. CCK is not just going to be responsible for bile flow, it's also going to be responsible for stomach motility, secretions from the stomach. It's also going to be helping us like designate that we have gut fullness. It's going to be playing back with the brain in some way so that we have like this gut-brain axis, gut-brain conversation, crosstalk that's going on. It's going to help facilitate that fullness that you feel after eating a high-fat meal. Now, I kind of want to go on a little bit of a tangent here. It's not completely unrelated, but it is a little bit beyond what you guys need to know for the class, but it's super important, right? In some way, shape, or form, when we consume medium chain triglycerides or coconut oil or MCT powders or MCT oils, we're actually going to take those shorter chain, medium chain triglycerides and send them straight through to the liver. When they get to the liver, they're going to go from medium chain triglycerides, and then they're going to be broken down into a molecule called acetoacetate. Acetoacetate is a 4-carbon molecule. This 4-carbon acetoacetate gets shipped out of the liver and gets converted in the blood to beta-hydroxybutyrate, which is a ketone body. Both acetoacetate and beta-hydroxybutyrate are ketone bodies. Within the blood, it's going to be pretty stable in this form of beta-hydroxybutyrate. When it gets to the target destination, which is like the muscle or the brain or some of the organs, the heart specifically, it will actually get absorbed into the cell because it's a lipid. It can go straight through the cell membrane, and it will be transported directly to the mitochondria where it converts back to acetoacetate, and then it gets converted into acetyl-CoA, and that acetyl-CoA can get plugged straight into Krebs cycle for ATP generation, which is super cool. So this is actually the mechanism by which a ketogenic diet works. We're just basically putting in a fake ketogenic diet. These are keto precursors. If any of you guys follow the supplement industry at all, you know that one of the big things that's hit recently is this idea of ketone supplements. You can simulate that by ingesting high doses of MCTs. You get a very similar trajectory, you get a very similar response inside the body, which is kind of cool, right? Now, how does all this differ from proteins? We know that there is a pretty big difference already just based off of the nitrogen, right? We know that the difference between carbohydrates and fats and proteins is that presence of that nitrogen group. Well, it's going to also kind of change the way that we handle some of the digestion and absorption. Within the stomach, we're going to have pepsin that's going to actually initiate that protein digestion. And we're going to have hydrochloric acid as well. So we're going to start to denature and start to break and open that protein. So when you denature a protein, you basically take and destroy the structure. So you're cooking the structure. If you've ever cooked eggs before, this is what you're doing. Literally the exact, very similar process. Not the exact, it's a little bit different, but it's very similar. So when you actually cook eggs, the reason that it changes structure, when you put it in the oil and it turns white and it actually turns a little bit solid, it's because you're denaturing the protein. You are literally literally breaking the protein as you look at it. So that's going to be what's kind of occurring within our stomach. We're going to be breaking it with acid, right? The same way that you break down meats with acid and that you can kind of like tenderize meats with acid, citric acid, lemon juice, lime juice, that type of stuff is the same thing that's going on here. And then inside of the small intestine, we're going to have continued breakdown with trypsin and chymotrypsin. And we're going to basically be taking those proteins, those denatured proteins, and breaking them down into small peptides and amino acids. And then we're going to transport those amino acids into the system. So it's it's a little bit different of a process. It starts a little bit early, and we're actually going to be breaking the structure down a little bit faster and a little bit differently, right? When it comes to proteins, we have primary, secondary, tertiary, and even quaternary structure. They have like all these different folds that they do to kind of create this big protein because they're like hundreds of thousands of peptide bonds long. So they end up folding into these goofy shapes. And what we're doing when we're breaking them down and denaturing them is we're opening them up, right? Opening them up. Kind of a cool process. So when we get down into the small intestine, this is where we're going to have trypsin really kind of start breaking down peptide fragments. We're going to actually couple the transport of amino acids with sodium in order to kind of get them out of the system. When those, sorry, it's cut off down here on the bottom. I was just trying to read ahead to see if it was it, and it is. One of the three events that occurs with those amino acids that actually get taken out of the small intestine and brought in to the bloodstream is we're going to convert them into glucose. So we're going to use glucogenic amino acids to actually be converted into glucose. We're going to be able to convert those into lipid with ketogenic amino acids. And we're also going to be able to have a direct release into the blood so that we can kind of increase the plasma-free amino acid concentration so that we can use it for things like nucleotide biosynthesis, anabolic natures, so building up muscle, that type of stuff. So this is kind of where we start thinking about fat-soluble and water-soluble vitamins. We've talked about how carbs and proteins and fats are getting broken down. The only thing that we're missing so far is what's going on with vitamins and minerals and what's going on with water, right? Well, our fat-soluble vitamins are actually going to occur predominantly as simple diffusion in the small intestine. And what's really nice about the fat-soluble vitamins is that they're actually co-absorbed. They're absorbed with the lipid, so that they're actually contained within the same micelle, within the same little breakdown product that the bile caused and they're brought across the cell membrane and into the system. Once they get into the blood, that's where they're going to be transported on chylomicrons and lipoproteins inside the blood. So it's a pretty simple process. It's a little bit easier. When it comes to vitamins, it's going to be a little bit similar, but a little bit different at the same time. So almost all of the water-soluble vitamins are going to have simple diffusion. They're just going to diffuse across the border in a passive manner. The only one that's different is actually going to be B12. Vitamin B12 uses endocytosis as its mechanism of entry into the system. The minerals are going to be different, right? The minerals are going to be something that our body struggles to absorb in a high level and absorb well. And realistically, a lot of this is because we maintain our mineral content within a very fine range, right? Like we're very specific with our range so that if our dietary intake falls, we can actually increase the absorption level to try to pull as much out of it as we can. So we're kind of fine. We're going through this with a fine-tooth comb. We're really trying to maximize absorption of these when we need them and not absorb them too much when we don't need them. In general, there's going to be kind of a couple of main factors that are going to be associated with the mineral absorption. We're going to have bioavailability. I think we talked about this last week. It could have been 2 weeks ago. But bioavailability has to do with how easily it can be accessed and actually grabbed as a human. How easy is it for the human to take it, internalize it, and use it, right? So certain substances are not going to have high levels of bioavailability. Certain minerals are not going to have high levels of bioavailability. Another big one is going to be transit time. How long does it take for it to go from the time you put it in your mouth to the time that it comes out of you? If we can increase that transit time, we can increase the likelihood of something being absorbed. This is where sometimes slowing down this process in that 24 to 72 hours can be nice. The amount of digestive juices that come out, the pH that comes out, all that type of stuff is going to be controlling this. But a couple of the big ones are going to be those first two, the bioavailability and the transit time. Then what's going on with water? Why do we have to consume so much water? Does it not just kind of like hit our mouth and go into our body somewhere? I did not plan to drink that water right there, but I like it. In general, our water absorption is going to come from pretty much our small intestine. We're going to have some of it coming from our large intestine, but the bulk majority of it is going to be coming from our small intestine, right? 72% of it's going to be coming from our small intestine. We're going to get a little bit from the distal segment, about 20% from the distal segment of the small intestine, and the remaining 8% is going to come actually from the large intestine. So there's going to be these kind of like 5 primary factors that are really going to affect our gastric emptying rate. Now, gastric emptying rate is super, super important because it's going to basically control how fast food is leaving the stomach. And the faster that food leaves the stomach, the sooner it's going to end up hitting the small intestine and the large intestine and start getting absorbed. And the slower things sit in the stomach, the longer you are to feel full, the longer you are to feel like you don't need another meal, right? So we constantly need to be adjusting this and playing with this so that we can try to maximize gut fullness, but also minimize the uncomfortable nature of having large amounts of food sitting within your stomach, especially if you're exercising. Like this, for an athlete, is huge. We need to make sure we're paying super close attention to gastric emptying rates of athletes pre, during, and post-exercise. So a couple of things that can kind of affect this gastric emptying rate is going to be how much food you're consuming, the calorie content of the food that you're consuming, the osmolality of the food that you're consuming, whether or not it's hot or cold food, whether or not it's hot or cold weather outside, as well as what the pH is. So what the acidity is of the food is all going to affect that gastric emptying rate for us. Now, we're also going to have to worry about exercise when it comes to gastric emptying rates. Exercise is going to actually cause a shunting of blood flow away from the gut and towards the periphery, towards the exercising muscles. So when we move blood away from the intestines and away from the digestive process and towards the periphery, we dramatically alter the speed at which we go through digestion. So as you increase in intensity of exercise, which is what's going on right here, we're going from rest to 3.2 kilometers per hour to 6.4 kilometers per hour. So we're increasing intensity or speed of exercise. Our gastric emptying rate is cut nearly in half when we go to 6.4 kilometers per hour. And you can see that as we decrease in that intensity, as we go from 6.4 to 3.2, 3.2, all the way back to rest, we actually increase the gastric, or decrease, we increase the gastric emptying percent, right? So we're able to actually increase the amount of food that is leaving the stomach. So why is this important? Why are we talking about this? Well, it's nutrition for performance class. So if you guys have ever tried to compete in anything with a full stomach, you know that it is awful. So if you try to slam something right before you go to work out or before one of your athletes goes to work out, they're gonna struggle with gastric emptying. They're gonna hold that within their stomach. So this is why those recommendations tend to be like 120 minutes to 90 minutes before exercise, sometimes as close as 60 minutes. But for the most part, we're trying to get that food in a pretty good ways before the exercise bout, so that we can make sure that it has left the stomach by the time the exercise bout or the competition begins. All right, that's the end of digestion and absorption. Next week, we're going to talk about bioenergetics, and then the following week, you're going to have your test. See you guys in class.